Hi, and welcome to the Master Methylator membership site. Today, I wanted to talk a little bit about how our genetics determine our best diet. We all kind of have, I think, a good basic understanding of some of this, but there's a lot of detail I think that might be very interesting to you. So I created a PowerPoint presentation, and I'm going to go ahead and share that now. All right, let me get this going. So, genetics and food, how genes influence your best diet. And to me, this is one of the more interesting topics when we look at our genetics about how to kind of live in the world and how to, our diet, if day to day you're eating the right foods that correspond to your genetics, it makes everything else easier. And in fact, it can lower your need for supplements, increase your energy, increase your digestive ability, just overall health. And though the knowledge, though, is a little bit tricky because things that we might not have thought were good for us can be good for us, and things that we thought were good for us might be bad for us. So it's really important to kind of understand you and your unique genetic makeup so that you can get the best possible diet for you. Let me get this going here. We're looking at proteins, carbohydrates, and fats and how they break down and then how they make energy or are stored as fat. So, most of us, there are some that don't need to worry about that, but most of us do have problems in having excess fat on our bodies, but not knowing what our best diet is. Is it carbohydrates? Is it fats? Is it proteins? What ratio is best for us? And everybody's got an opinion. There's the keto diet, there's the protein carnivore diet, there's the low carb diet, there's the just name it. Every variation you can think of, and they all work for somebody, or nobody would have written, or they wouldn't have written the book. But really understanding you and your genetics and your best dietary ratios is super important. When I discovered mine, I had been eating for most of my life like a high protein, low carbohydrate diet. And didn't, I mean, initially I could lose a little bit of weight, but it would just come right back on me. And I just never understood because I read the book Protein Power many years ago, and I really thought that was the best diet. It was compelling. Every diet book you've ever read is probably compelling on some level. So it's really difficult, to figure out which is your best diet just through, left brain reading of a book. So when we look at our best diet, we're looking at certain genes that will allow us to turn proteins into amino acids correctly, or carbohydrates into glucose, and then ATP or energy instead of storing it as fat, and then triglycerides and fats being turned into fatty acids and then converted into ATP or ADP and then ultimately ATP or energy. So let's find out how you can tell what your best ratio is. So these are some of the genes that are involved in difficulty with processing fats in the diet. And they include the PEMT, which you're probably familiar with. It's up in the methylation cycle. It's a small methylation pathway up there that converts phosphatidylethanolamine into phosphatidylcholine. And phosphatidylcholine is very much important in processing fats in the diet. In fact, it can lead to non-alcoholic fatty liver disease if we have a genetic variant there. The DIO2 involved, the ACATs, which we're real familiar with, where cholesterol can't be broken down into bile. And so it causes a high levels of cholesterol. Usually recommendations there are a low-fat diet. MMAB processes fats into the electron transport chain. And then the SLC22A, SLC22A5, 4 and 5, actually allow carnitine to get into the cell so it can burn fat as a fuel. So genetic variants there definitely going to cause issues with using fat as a fuel. And then the NDUFS gene, very much involved in how we can break down fats into energy. The FADS gene, the IL-12A AS1, those two are highly susceptible, highly associated with difficulty in breaking down fats into energy. These are some of the genetic variants associated with difficulty with carbohydrate diets, and these are going to lead to high blood sugar, high insulin kind of issues in the body. And so, some of these are going to look familiar, like the N-Duphus, of course. Again, it just stops everything right before the electron transport chain, and so everything is sort of stored as fat. So it's going to show up in several categories here, but the rest of these are all associated with difficulty. Any block in that citric acid cycle is going to cause glucose to stay high and insulin to stay high and increase insulin resistance. So these are all associated with high insulin and insulin resistance kind of conditions. These guys are involved in high protein diets. So, that we're looking at, again, MMAB, it's shown up like 3 times here. It will block the production of in the electron transport chain, but right before it in the Krebs cycle, it'll block Krebs cycle production. The TPHs are going to cause issues with tryptophan. So this is tryptophan hydroxylase. So if you can't turn tryptophan into serotonin because of that genetic variant, then foods that contain tryptophan, which are proteins, are not going to be your best friend. So, but that's true of both the TPH1 and the TPH2. And then the TH is the tyrosine hydroxylase. Again, the only place that you find tyrosine is in protein. So it can be contraindicated for those people with genetic variants there. And the HFE gene is associated with hemochromatosis, so high blood levels of iron. And, you'd want to watch the protein, not just the protein in meats, but anything with high iron in it, including red meat and spinach, and other foods that have high iron would be contraindicated for that particular genetic variant. And then the VDR variant is associated because it's involved in how we turn tryptophan and tyrosine into serotonin and dopamine. So a genetic variant there is going to lead to more of a vegetarian susceptibility. And then the MTRR and MTHFR, the genetic variants in the methylation pathways, can lead to issues with breaking down our protein. So alcohol, these particular genetic variants, the sulfur transferase, so anything with sulfur in it, including wine and beer, is going to be a problem. But the rest of these are either aldehyde dehydrogenases or alcohol dehydrogenases, and they're very much involved in how well we can break alcohol down into aldehydes, and then those aldehydes down into inert substances out of the body. Caffeine, these particular genetic variants are going to have issues with breaking down caffeine. The COMT gene, as you know, is associated with high levels of adrenaline, and whenever you drink caffeine, it's going to increase your adrenaline levels. The NAT1, NAT2 are directly involved in how we break down caffeine. So genetic variants there are going to cause a person to be really jittery when they drink caffeine. The ADORA2, the ADA, the ADH7, those 3 are also involved in how well we process caffeine. So some people do really well on caffeine if they don't have these genetic variants. I happen to be one of those, and I'm happy about it because there are a lot of other things that send my body the wrong way, and caffeine has become a good friend of mine. But I don't have genetic variants in any of these genes, which is kind of handy. And as far as fermented foods, we have the DAO, HNMT, and the MAOs. And of course, these are all up in the histamine pathways in the upper left-hand side of our pathway chart. So they are going to lead to, fermented foods generally are high in histamines. And so they're going to have people with these genetic variants would be better off if they avoided fermented foods, because that's going to lower their histamine levels. And we'll talk about histamine, other histamine foods too, but these specifically are more contraindicated for fermented foods like kefir and yogurt, kombucha, those kinds of things, kimchi. Not a good idea for these particular people. And these are things that people will tell you, oh, this is the best food, you should eat fermented foods, it's really good for your gut flora. Well, that may be, but it also is contraindicated if you have these genetic variants. These particular food allergy genes, the HLAs, are human leukocyte antigens, and they're highly associated with certain food sensitivities, including like peanut allergies, gluten sensitivity, shellfish, the things that are like anaphylactic shock kind of reactions can be in this category, but also this is highly associated with autoimmune conditions. So you might see cross reaction between foods and body parts and other targets of autoimmune conditions in the body. So the HLAs, you got to be particularly careful about, and it's nice that we have muscle testing because you really need to get in there and muscle test to see what foods these people are reacting to. And this is a case where, especially if you see it on their report, if you see the HLA variants on their report, you'll want to use the HLA vials, the 4 vials that we've talked about quite a bit, the HLA, the MCH class 1, MCH class 2, and the cytokine vials, and put those on the body and then screen the foods, the food kits, because without those screens on there, you might not find the HLA reactions to food allergies. So these guys, definitely watch for these on your My Happy Genes reports, because these are the tricky people. These are the ones that are going to have some bizarre food reactions that you can't figure out, because they're not going to muscle test weak on these foods until you put those challenge vials on there first, or screening vials. These particular genetic variants highly associated with gluten intolerance, especially the HLA-DQA1, the CCR3, HNMT, and DAO, those are the ones up in the histamine category, the RGS1 and the MTHFR, that mother flipper. What happens with gluten intolerance, the T lymphocytes invariably recognize gluten peptides that are presented by HLA-DQ2 or DQ8 molecules. So these are the other 2 gluten intolerance genes. This results in the lymphocyte activation and initiation of inflammatory reaction and autoimmune responses. So we want to definitely watch for those on a report, HLA-DQ2s and DQ8s, tricky little boogers. Celiac disease, when you have these genetic variants, can lead to some interesting symptoms, including brittle nails, acne or eczema, intestinal diarrhea, bloating, constipation, infertility, miscarriage or early menopause, ulcers and tooth enamel erosion in the mouth, joint and muscle pain and swelling, stomach pain and nausea. It can also lead to lactose intolerance, anemia, dizziness, migraines, depression, low vitamin D, and chronic fatigue. Low vitamin D because of the absorption issues. So we look at, so right in the center of these 3 circles is the autoimmune disease. So it's not always just genes. It's not always just the HLA issues. We need to also look at vitamin D deficiencies and leaky gut conditions that can lead to the expression of the autoimmune disease, along with the genes, and then our environment, including gluten, grains, nutritional deficiencies, viruses, bacteria, etc., can lead to this activation of autoimmune diseases. And, one thing to mention about gluten, it's not always the gluten that's a problem. When you test somebody on a piece of bread, let's say, it's not always the gluten. Sometimes it's the glyphosates, or it's the way it was processed, or it's aflatoxins or molds, or how the grain was stored, etc. So there are lots of reasons why somebody can be reacting to wheat or rye or oats or barley. It's not, gluten gets a bad rap, but it's not always gluten. Histamine intolerance, the genes involved would be the HNMT, MAOB, DAO, the mother flipper, MTRR, MTR, DHFR, and MTHFD. And you can see, these guys, these are the methylation genetic variants up here. These are the 5 bad boys when it comes to methylation, and the reason that they're implicated in histamine intolerance is that low methylation can lead to histamine building up in the body. It's one of the main indicators of low methylation is high histamine, high blood histamine. So the most common symptoms of histamine intolerance include migraines, digestive symptoms such as diarrhea, flushed skin, hives, worsening eczema, congested, runny or itchy nose, red, itchy, or watery eyes. And this is the list of high histamine foods. I'm not going to read this to you, but I just wanted you to have the list here. And don't forget alcohol. It's way high in histamine, fermented beverages, fermented foods, and cheese, etc. So it's kind of a big list. Variants in certain genes can increase the inability to break down histamine efficiently. Those who have difficulty producing methyl groups, such as the MTHFR gene, will have more difficulty breaking down histamine. Imbalances can lead to inflammation, agitation, anxiety, and other brain imbalances, along with the traditional symptoms of high histamine, such as allergic reactions to pollen, etc. So it's going to increase the load, the histamine load, when you eat these foods. And I just want to point out this agitation, anxiety, highly associated with high histamine levels. And there was a book I read many years ago called Brain Allergies, and it was just eye-opening, stunning how certain food allergies can cause mental health issues like agitation and anxiety, but depression, bipolar, schizophrenia, highly associated with food allergies as well. These are the genes associated with difficulty in intermittent fasting. So when I look at this, I look at how well can a person maintain blood sugar? How well do they turn glucose into energy? And if there are blocks along that pathway, and these are, as you can spot them, they're all blocks along the Krebs cycle and the electron transport chain. So what that means is you're going to run out of fuel pretty quickly. So for intermittent fasting for these folks, it's difficult because they'll go into starvation mode and their body's going to make it worse. They're going to make the body hold on to calories even tighter because you're already starting with like an empty tank, and then you empty it even more by skipping too many hours of meals, and these people are not going to do well. Their brain kind of shuts down. They get tired and sleepy and can't function correctly until they get some fuel in there. So it's going to be difficult for them to go too long on intermittent fasting. That doesn't mean they can't do like a 12-hour intermittent fast. Most people can handle that, but how long you can go really does depend on your genes. So if you go to 13 hours and you're like, oh my God, I can't function, you know you hit your limit. So pay attention to your body. But some people can go 16 hours, 18, more, and they're fine, or they're fine on one meal a day. I'm not one of those people. I don't know about you guys, but for me, I have to have consistent meals to keep my energy up because I have some genetic variants in these pathways, especially the SLC, 22A4 and 5s, the NDUFAS, and the MMABs. They're going to be highly implicated in this. Intermittent fasting, though, and calorie restriction have been shown to extend lifespan and increase resistance to age-related diseases and improve the health of overweight humans. It's been shown to enhance cardiovascular and brain functions and improve several risk factors for coronary artery disease and stroke, including reduction in blood pressure and increased insulin sensitivity. So it's good stuff. It's just not for everybody. So let's look at lactose. We have MCM6s and LCT. These are the only two genes I've seen so far associated with lactose intolerance. It's estimated that lactose intolerance genetics by ethnicities from most intolerance to least intolerance. Looking at South Asian, East Asian, these are the most intolerant Ashkenazi Jewish, African, Latino, and the non-Finnish Europeans are least likely to have issues with lactose intolerance. You can see a worldwide prevalence of lactose intolerance in recent populations. It's fascinating because, I don't know why. Looks like the southern latitudes, the southernmost countries are more lactose intolerant than are the northern countries. And there's probably a reason for that. I don't know what it is yet, though. MSG, these two genes, the GAD1 and GAD2, are highly associated with MSG intolerance, or the inability to break glutamate down. Glutamate is a neurotoxin and a stimulant that's toxic and highly addictive. It causes a relentless firing of neurons that cause cravings, food addictions, obesity, and its complications, inflammation, autoimmune disorders, and various cancers. So it's a nasty gene. It's definitely something you want to watch out for, that GAD, and it is on the danger warning list when we go into methylate, because sometimes we can push up the production of glutamate without realizing it, and then we can't break it down, and we're going to create all kinds of health issues for somebody if we're not paying attention. The nightshades, the main gene here is the BCHE. It provides instructions for making pseudocholinesterase enzyme involved in the breakdown of certain drugs, including muscle relaxant drugs or anesthetics. Pseudocholinesterase also helps protect the body by breaking down certain toxic substances before they reach the nerves. These substances include certain pesticides, poisons that attack the nerves, and specific natural toxins, including a compound called solanine found in green potato skins. And it's an interesting gene because this brings up a point that you can't look at a gene and go, this is a bad gene. This is a bad gene to have. It has, some effects in the body, some positive effects. So it's always a trade-off with genetic variants. So salt genes, the ATP2B1, the BCAT1, and FGF5s can cause salt intolerance. Salt can be highly addictive. Salt need and cravings may be linked to the same brain pathways as those related to drug addiction and abuse. Thought that was an interesting little tidbit there. And tryptophan, the genes involved in tryptophan intolerance are going to be the MAOs, of course, and the TPHs. So for these people, they're going to have a lot of difficulty breaking down tryptophan, and tryptophan is found in things like wine and cheese, and of course turkey. Backing up a little bit. So tryptophan intolerance is a nasty, nasty thing. And if you have these genetic variants, it can back tryptophan up into kynurenine and quinolinic acid. And those are highly neurotoxic. So for people that have these genetic variants, a low protein diet is going to be more indicated for them. And this is true of me. I've got several variants in a lot of these genes. And so tryptophan is not my friend, and I've found that out kind of the hard way. Chocolate's in that list too, but I have to kind of stay low in my protein consumption because it can lead to my tryptophan levels building up too much and creating migraines, joint pain, brain chemistry imbalances. It's kind of a nasty thing. I think I've talked to you all about that before. Soy, these particular genetic variants are associated with how well we break down estrogens. So the COMT, UGT, SULT, CYP1B1 are going to cause some problems with soy as it is a phytoestrogen. Sulfur genes, we are familiar with these, the SALT, SUOX, and NDUFAS too, because that NDUFAS can lead to a buildup of reactive oxygen species and a buildup of sulfur, because the sulfur should normally be used to create energy or ATP, but that genetic variant can cause a backup of sulfur in the body because it isn't using it to make energy. Sulfur can lead to imbalances in brain chemistry, anxiety, allergic reactions, lung problems, digestive distress. Foods that are high in sulfur include wine, beer, eggs, onions, garlic, broccoli, cauliflower, and coconut products. Adverse symptoms may include flushing, dermatitis, urticaria, hypotension, and diarrhea to life-threatening anaphylactic and asthmatic reactions. It's a sneaky one, and I know you've probably run across sulfur reactions before as well. That coconut, a lot of people just can't get it that MCT oil is coconut. That's medium chain triglycerides. Yeah, very, very healthy for some people, but if you have these genetic variants, not for you. And again, that just brings home that point. There's no one size fits all or one food is good for everybody. Back to tryptophan again. Those persons with MAO and TPH genetic variants have difficulty processing tryptophan as serotonin and are highly susceptible to mood imbalances, irritability, and aggressive behavior. So avoiding chocolate, wine, hard cheeses, turkey, and spinach are indicated. In addition, spicy food can increase the irritability in those with MAO variants. We associate the MAO variants as the warrior gene, but also in Ayurvedic medicine, it would be a pitta imbalance, and that spicy food can add to that imbalance. And then tyramine, there's the MAOs are very much associated with tyramine intolerance, PNMT and the CYP2D6 genes. And if you look in that middle pathway, the brain chemistry pathway, you'll see tyramine sitting there in the middle of the tetrahydrobiopterin pathway, because it's broken down also into serotonin. So for these people, wine, beer, chocolate, aged cheeses, and fermented foods, leftover foods, grapes, and cured meats, and more are going to be an issue. So definitely testing them. If you see tyramine go weak on them and you have these genetic variants, definitely caution them to watch these foods because they can really add to symptoms pretty quickly. Tyramine intolerance. Certain genetic variants can lead to something called tyramine intolerance, which can cause elevated blood pressure and heart rate, heart palpitations, migraines, mood changes, nausea, and vomiting. In addition, some people will experience early waking syndrome. So if you have a patient who's waking in the middle of the night, around 3 o'clock, often it can be due to a food sensitivity, but tyramine specifically is implicated in that kind of early waking syndrome. So that's my story on the foods. I hope this was helpful to you. It's going to be a short session tonight, but I think I'm hoping that you pay a lot of attention to the diet report on My Happy Genes, because it can really tell you a lot, and it can be very helpful, especially for those people that you don't seem to be getting anywhere with. You're doing all the methylation correctly, but they're going to be disrupting the program and sabotaging it by eating the foods that aren't congruent with their particular genetic makeup. So hopefully you'll pay a little more attention to that. And I know you all are testing the allergy kit, and that's cool. And I would actually encourage you to test it before you put the methylation program next to somebody, just to see. Look, this is probably a good idea for you to avoid these foods because genetically they're not going to be your friends. It's good to identify those before you cover them up with the methylation program. And here's another really cool thing that I just discovered. Should have discovered it. It's kind of a duh. Is using the IgG vials as a screen as well. So if you put the IgG vials, IgG, IgE, IgM, and IgAs on the body and then test the foods, you can find an immunoglobulin response that maybe wasn't going to show up without that fine screen. So you can test these in two different ways. I haven't tried them together with the HLA vials. That would be interesting to see if you had to put all 8 on those. In fact, we could make one vial, out of all the 8 of those, like a allergy sensitization vial or something, and then you can check the foods. That would be fun. Maybe I'll play around with that and see if I can come up with like one vial that would kind of be the screen that you could put on people. But I have found it to be revealing. Sometimes they're not reacting to an HLA issue, but they're actually having an immunoglobulin response to a food. It's different than an allergy. It's more like an immune response. So it's going to muscle test differently. So it's just fascinating to see that. So. All right, so that's my story. I'm hoping that gave you some good little tidbits and some good information. And I'll see you next month. We're going to have Dr. Harry Eidner on, and he's an amazing guy, really the most knowledgeable on blood work of anybody I've ever met. And let me show you the book he wrote. I use this book all the time. It's Balancing Blood Chemistry with Nutrition. More than just a bunch of numbers, making sense of blood chemistry results. And it's so cool because you can look up a blood chemistry, let's say serum creatinine. Here's one in here. It'll show you what it is, related tests, reference range, optimal range, possible panic range, and then common causes, and then nutrient recommendations here. So you can look at somebody's blood work, even if you don't know what you're doing, just get in here and look up that particular blood work parameter, and if it's out of range, you can kind of get a sense of what to test or why it's out of range and what to do about it nutritionally. And he's an amazing man, and he helped me design the My Happy Genes blood work panel. And so he's going to talk about the association between genetics and blood work and how you can spot genetic variants through blood work and why we picked certain parameters to put on our My Happy Genes blood work panel. So I hope you don't miss that one. I think it's going to be really enlightening and a great time. He's going to give us also a discount on this book if you're interested in getting it. So wait to get the book until you listen to the webinar. So he'll give us a special deal on getting that book. Alrighty, so thank you so much for coming and hanging out and viewing the video. And I hope to see you next month, first Thursday of the month. I think that's September 6th. I'm pretty sure, 6 p.m. See you guys then. Bye.